Okay, I'm going to tell you about some work we undertook between 2010 and 2016, investigating the effects of 1080 on forest birds. Now, at the time we started this work, it's hard to think back so far because it feels like a long time ago now, but we were quite unsure what we were going to find. We knew that some birds were killed by 1080 and that birds benefited from rats and stoats being killed by 1080, but we had not much idea about what the balance of the gains and losses was. So this is what we were setting out to find out. Now, in the tradition that Josh started and Susan has continued, I'm going to, if I can get my next slide up, so you don't have to wait impatiently for the whole talk to hear the punchline. Here in verse is the guts of what we found. 1080 is good for birds in the wood. It's great, hey? You can clap and cheer if you like. Now, so you probably don't need to hear the rest of my talk because that summarizes it all, so you can all bugger off. Okay, so we worked in two places mostly, in Southwestland at Lake Paringa and in Tennyson Inlet in the Marlborough Sounds. At both sites, we counted through multiple 1080 operations, hoping to detect increases and decreases in abundance of a variety of species that were associated with, well, the changes were associated with 1080 use. We didn't use people to do the bird count. We used digital audio recorders. We used people to carry the bird counting machines around. We collected a hell of a lot of bird counts, and it's taken us ages to process and analyse them. And I'll describe what we've found. But a bit more description of the study first. So our study site in Southwestland was centred around Lake Paringa, which is a funny little U-shaped thing in the middle. I wonder if I can, how do you make a thing go like that? There we are, there's Lake Paringa. There's 35,000 hectares, and it was divided into three blocks. The Whakapohai block, the southern one, received 1080 operations at about the time of the beach flowering. This is before the rat and stoat numbers rose and just before the kaka breeding. It was sort of designed around maximum benefit to kaka. Now, on and off throughout the rest of this talk, I'm going to talk about nip it in the bud 1080 operations, and these are 1080 operations, ones that happen at about the time the trees flower, and they're intended to get rid of the baddies before their numbers rise. The Paringa block, the middle one, received 1080 operations after the beech seed had fallen and before the rats and stoats reached their peak density. So this was aimed to have maximum benefit to small rat-sensitive forest birds. And throughout the rest of this talk, I'm going to talk about this as being food glut 1080 operations. So these were 1080 operations that occurred when there was lots of seed on the ground, the food glut. And there was a non-treatment block at the north where nothing happened. During the time we worked there were 3 10 operations in each of the treatment blocks, and there was one at the Paringa block before we started our work. So this is how it went. So this was a food glut operation in 2029, just before we started. Then there was a nip it in the bud one in 2010. A food glut in 2011, nothing in 2012, 2013, another one of these nip it in the bud ones, followed by a food glut one, nip it in the bud, and this last one in 2016, another food glut operation, but actually wasn't much use to us because we stopped working that at the end of that year. This is how we arranged our bird counts. So they were in 6 circles of 4K diameter, and we had 50 bird counting stations placed at random each side of the circles. The circle to the very northeast, I suppose, is kind of outside our study area. We couldn't resist doing that because we wanted to see what the birds were doing in a completely different forest type. There's no beach up there, but I'm not going to talk further about that one. So we also had a bunch of tracking tunnels sort of matched up with each circle. So there were 9 lines of 10 tunnels with which we monitored rats and mice and stoats. And as well as that, we ran wax tags at sort of irregular intervals throughout the study to keep tabs on what the possums were doing. There was a bit of a sort of flaw in our study design in that the 3 blocks that I chose at the beginning because I thought they were really, really well matched turned out to be not that well matched. So this map is of the forest types, and the dark green stuff is pure beech forest, the pale green is beech podocarp, and the brown stuff is sort of podocarp beech with lots of remu in it. So you can see there are 2 northern blocks, the non-treatment and the Paringa block, were really quite well matched. They had a very similar match of forest types, but the block to the south, the Whakapohai block, was quite different, and it was much, much more beachy. Another important feature of our study which was kind of unfortunate, was that the two southern blocks had a history of treatment. They'd both had several 1080 operations before we started doing the study. So we didn't kind of get the before-after treatment control that we would have liked. It was really just a treatment control study. This is a view of the country where we did most of the work. You can see Lake Paringa down there. This is the Paringa River flowing here. Most of our work was undertaken in this sort of low hilly country out here because we didn't want the steep mountainous stuff. And it's all east of the Alpine Fault. The Alpine Fault runs through our study area roughly along that red line. Enough of Southwestland. Our study site in Tennyson Inlet looked like this, much smaller, only 8,000 hectares, divided in half. We had a non-treatment half and Mount Stanley, the bit that got treated. We didn't have great control over when the treatments happened actually in either block, but in this one it was particularly unfortunate, really. So we were studying birds madly for years, and we didn't get any 1080 operations until 2013. And then we got another one in 2014, and then no more. So it wasn't an ideal design. Here's where our bird counting stations were. Same sort of story, except the circles are smaller. These are only 2K circles, but we've still jammed 50 counting stations into each one. The country in the Sounds is much, much rougher than the country in Southwestland. So actually having the bird count stations much closer together was a bloody good idea. We also had a bunch of tracking tunnels to keep track of rats, stoats, and mice. And same as in Southwestern, we intermittently ran wax tags to keep tabs on what the possums were doing. Now, we only had one set of sound recording gear, one bunch of digital audio recorders, so we couldn't run the two studies at the same time. So we ran the Tennyson Inlet studies every year in November, December. We had all the gear out recording, and then we moved all the gear down to Southwestern and did the work down to Southwestern from January to March. This is just to show you how they were laid out. So this is an example of one of our circles in which we counted birds, and that's where we had the counting stations. And I wanted to talk about the advantages of doing this with audio recorders rather than doing it with people. If we'd done it with people, we'd have had access to our blocks from these two road ends, and this is where we indeed walked into the blocks. And if we'd done it with people, we'd probably have tried to do it earlier we'd tried to do the bird counts in the morning. With the digital audio recorders, we only recorded in the morning, and obviously there's huge change in bird abundance, well, bird noise rather, with time of day. So you often do bird counts in the morning. If we'd just done bird counts in the morning, this is about as far as we'd be able to get into our block and be out in time for lunch. And so this is what we might have been able to achieve with our bird counts, a bunch of bird counts along little lines like that. But instead, by not being confined to doing it in the morning and not using people to actually do the counts, we could walk all over the blocks. We could put the last ones out at 5 o'clock at night and then run home. We left the recorders out for a couple of days and used the recordings that occurred on the fine days after we put them out. Analysis of the recordings was a bit of a beast. We used people, not AI, and we had the people listening to recordings and looking at sonograms at the same time as we listened to them. And at first we did it in a kind of lab environment. We had people sitting around working together and comparing notes, and we hoped that eliminated some of the inter-observer differences you get when you're doing bird counts. We also did the counts in stereo. So each digital audio recorder had two microphones, and they were placed 30 meters apart in the forest. And this gave us a kind of stereo effect when you listen to them. So as you were listening to the calls, some birds would sound like they came up from the far right and some from the far left. So you could actually count the birds using the stereo effect. We didn't have to do just presence absence. And also using two mics is going to enable us to do some spatially explicit analysis using some tools that Murray Effords figured out, where we can use the different signal strengths at the different mics to get a sort of proxy for distance away from the microphone. I haven't done that yet. We use different counting metrics for each of the different species. So for some species, we simply counted them. We could listen to the whole recording and say, I heard 6 kaka. But for other species, particularly bellbirds in southwestland, they were so common that just counting them was hopeless. You couldn't keep tabs on all the birds. So for those birds, we chopped the counts up into smaller sections and recorded presence absence in the smaller sections. So for bellbirds, I'm struggling a bit to remember, but I think we chopped it up into 10 or 21 second bits and scored presence or absence in the 1 second bits. But anyway, for each species, we had a sort of tailored metric that we used. So how many counts did we process? We recorded 110,000 5-minute counts. We did them for 5 minutes to make it a bit comparable with 5-minute bird counts. But many of these were recorded, in an hour in the morning. We didn't want to analyze them all, so we only looked at 36,000 of them, and of those, 29,000 were processable. That it wasn't too noisy, too rainy, too windy, something like that. So we processed 29,000 counts. Now, they were roughly 60% from southwestland, 40% from the Marlborough Sounds, and they were 50-50 night and day. We did nighttime counts as well as daytime counts. It took 8 person years of processing to get through those counts. Yuck. Now, the statistical analysis we undertook, I'm just going to go blah, 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 blah. We used generalized linear mixed models to do this stuff. But what I want to talk about is that we had a bunch of nuisance variables. There were things like background noise, microphone sensitivity, because we used a large number of potentially different microphones. There was time of year, because it often took us a month or two to get through each of the counts. There was time of day. They weren't all done exactly the same time of day. There was altitude and there was forest type. So all these things potentially buggered up our analysis and had to be taken account of when we did our generalized linear mixed models. The variables of interest that we had in the models were the time since 1080 and whether it was a nippet in the bud or a food glut 1080 operation, and the beach and remu masts. Now, it would have been rather nice if our data analysis had gone like this. So we had a bunch of 1080 operations in treatment and non-treatment blocks, and we could have just, so counts, and then we could have just fitted a line like that. And it would have been great if the treatment lines had gone up and the non-treatment lines had gone down. And that they were significantly different. But with lots of noise in counts, we all know that bird counts an incredibly noisy process. I don't mean the noise from the birds, I mean the statistical noise from variable counts. And with only 7 years of counts and only 3 1080 operations, and in Tennyson we only had 2 1080 operations, and they were very close together. So the chances of getting a sort of compelling result out of this stuff were a bit slim. So instead of fitting lines, we manipulated our GLM analysis so that we looked at between-year changes in counts in the treatment and compared them with the between-year changes in the non-treatment. So the sort of process kind of goes like this. So imagine if we just fitted a line and these counts were above the line, were sort of positive, these counts were below the line and sort of negative. But then if you look at this one count here, you see that even though it's below the line and kind of a negative impact, it's increased substantially. Count, the birds went up in that between those 2 counts, and it was just after a 1080 operation. And then if you compare it with what happened in the non-treatment, the non-treatment went down. So even though if you'd done the traditional, straight line analysis, this would have been below the line and a kind of negative. In fact, the bird count rose substantially, and at the same time went down in the treatment. I'd say that was a positive effect, which might be associated with 1080. So anyway, as I said, we manipulated our analysis so that we examined between-year changes in counts rather than just straight lines and compared them, the treatments and the non-treatments. Okay, some results. I'm going to show you just a few species because I've only finished the analysis of a few species. I've been, I was up last night trying to add a few more species into it, but didn't achieve that. But there's going to be more to come as we process more of this. So this is Korimako bellbirds at Paringa. I'm going to show you a few graphs like this one with years across the bottom, bellbirds or whatever species it is up the left. And I'm going to have different coloured lines for the non-treatment. The non-treatment's always going to be red, and the treatments are going to be blue and or green. And then at Tauranga we had 2 treatment lines, and Tennyson we only had 1. And the arrows are coloured to match the kind of 1080 operation they were. So the blue ones are food glut operations, and the green ones are nip it in the bud operations. Now, I don't think you're going to get much out of staring at these graphs because there's a whole lot of confounding effects going on. There's weather, altitude, forest type, time of day, time of year, all that kind of stuff. So the analysis of what's gone on really has to be statistical rather than graphical. But I had put something up while I was talking, so you're stuck with these graphs. The first thing to notice, though, is that bellbirds are about twice as abundant in the treatment blocks as they are in the non-treatment block. And remember that at Tauranga, the 1080 operation started long before we actually started counting them. So what we imagine happened here is that bellbirds increased in abundance once the 1080 started, and it's just roughly stabilized. So we've got higher numbers of bellbirds in the treatment than the non-treatment. But I still did the statistical analysis, and it showed that during the food glut times 1080 operations, the bellbird rose. Hooray! So Korumako at Tennyson Inlet, we've got a couple of 1080 operations, and this one actually, if you look closely at the graph, you can kind of figure out what might have gone on. How do you do this? Oh, there we are. So after the first 1080 operation, which was a nip in the bud one, the birds in the non-treatment declined, and they also declined in the treatment block. So really, that first 1080 operation didn't make any difference. But after the second 1080 operation, the birds in the treatment block rose, and the birds in the non-treatment block declined. So there's a good chance there was some sort of treatment effect there. And if you did the stats, it said that the food glut time 1080 operation has a positive effect on bellbirds. Hooray! Am I going for time? Oh, yeah, I'm good. Okay, next species is Kaka at Tauranga. There was almost no Kaka at Tennyson Inlet, so you only get results from Tauranga. Like bellbirds, Kaka more common in the treatment than non-treatment blocks, and remember that 1080 started before we started. But from Kaka, we could get no compelling evidence of a relationship between the timing of the 1080 operations and what Kaka did. At the same time that we did this counting study, we did a rather detailed study of which Jason did most of the work. We had radio tags on the birds we found nests, we monitored their nesting success and their survival. And we've actually published this stuff, it's out in the literature, and the intensive study showed significant benefits to survival and productivity of 1080 operations, and that if the 1080 operations were carried out at regular intervals, then kaka populations would increase. So it kind of seems like same old story. Chances are kaka numbers had increased in the past through the repeated 1080 operations that occurred before we got there, but that either the kaka abundance have reached a new equilibrium, or that the counts are so noisy and the birds are so mobile that trying to detect changes with bird counts was hard work. Anyway, Miro at Tauranga, tomtits, same old story, much less abundant in the non-treatment block, more common in the treatment block. We suspect that's because of the previous 1080 operations. And when you do the stats, it seemed that tomtit numbers rose using both kinds of 1080 op, both the nippet and the bud and the food glut ones. We did the same analysis at Tennyson Inlet, and we couldn't detect any impact of the 1080 operations. And at Tennyson, there's obviously no treatment control abundance difference because they started at the same time. I got sick of graphs, so now I'm just going to show you pictures and tell stories. So like kaka, there's almost no kakariki at Tennyson Inlet, so I've only got results for Tauranga, and kakariki were less common in the non-treatment than in the 2 treatment blocks. And we found they were declining in the absence of treatment, but they increased a lot after food glut 1080 operations. They increased a little bit after nippet and the bud 1080 operations. And they also increased substantially when there was a mast. Parakeets eat beech and remu seeds, so they do quite well out of it. Next one's rifleman, TTT Ponamu. No obvious difference between the abundance of these birds in the non-treatment and treatment blocks at either place, but there was a big altitude effect. Titi Panamu are much more common up higher than they are down low. And at both places, well, they increased with nip in the bud operations, and at Paranga, they also increased with the flu 1080 operations. Like Kaka, at the same time that we did this counting study, we also had a detailed study going on, banding survival, nesting success. And my colleague Claire Kilner has written this stuff up, and it's about to be published in New Zealand Journal of Ecology. We kind of got a very similar result from the detailed study and the count study. South Island robins, which are apparently called kakaruai, we could find no obvious relationship between 1080 and kakaruai in counts. And we only did this work at Tennyson because there were no robins at Palinga. Once again, at the same time we did this bird counting study, we also undertook a detailed, intensive study of nesting success, and we found that nesting success increased after 1080 operations at Tennyson Lynette, but the effect was very short-lived, and it was kind of overwhelmed by Susan's rat bounce effect. And so the benefits of increased robin productivity in the year or two after 1080 operations tended to be undone by the rise in rats during the rat bounce that happened in the subsequent years. I blame Susan for that. Okay, so we're sort of done with it all. So this is the where we got to, just summarized all those little arrows going up and down. So for all those species except kakaruai, we had a benefit of 1080 at least one of the sites. But there were differences between how the birds benefited between the nippet and the bud timed operations and the food glut timed operations. And there was considerable evidence, if you like, from Paranga that much of the benefit from 1080 had already occurred before we started doing the study. Yep. Okay, for some concluding comments. The power of our study came from the very large number of counts we did. But it was very expensive, and all of us, we had a big team here doing it, and we all got absolutely sick of it, and we wouldn't do it again. Large bird count studies like this really need artificial intelligence to do the analysis rather than people sitting around doing it. But you don't want to believe the folk telling you that AI is ready to go. At the beginning of the study, I asked around the AI folk about whether or not I could do the analysis using AI instead of people. And they all said, oh, yes, you could, or at least you need just to wait a couple of months and it'll be ready to go. And it was just around the corner. And here we are, 14 years after we started, and it's still just around the corner. So, if you were going to use bird counts for studies like this, I'd be inclined to use them for sort of low annual cost with smaller number of counts, but do the studies for a very long time, and you might see the trends come out of it. Another important sort of finding, or at least hint from our study, is that for most species, the benefits of 1080 occur quickly. Imagine a bird like a bellbird suddenly benefiting from increased pest control that can probably produce 5 or 6 chicks a year. So if things get good, the population increases rapidly, and within a couple of years, it's reached its new carrying capacity. If you study if you start counting your birds before the pest control starts, that's going to be great. But you're still only going to be able to detect increases for a couple of years, because chances are they'll only increase in abundance for a couple of years. And if your bird counting is a noisy technique, which it always is, then you're going to have a pretty powerful study to detect changes in bird abundance over just a couple of years. And of course, if your study starts after pest control, then you perhaps can only expect to see the population stay stable. You'll be looking to see that doesn't decline. Maybe it's not going to go up. I've been calling this the Dave Kelly effect, because this is sort of something that Dave pointed out to us a few years ago. So this bird count study showed that most of the native birds benefit from 1080. And remember, we started this because we didn't know whether they benefited or from it. And so this showed that they did. I haven't finished doing the analysis of all the birds. That's going to happen, and we'll publish this stuff quite good. So going back to where we started, 1080 is good for birds in the wood. Thanks.
Thank you very much, Graeme. That was superb. And from all the audience reaction, I think you can tell that that was really thoroughly appreciated. We've got time for probably one of these two questions. Nathan Whitmore asks you, Graeme, apologies if you mentioned this, were the treatments randomly assigned?
Were the treatments randomly assigned? No, they were assigned by others. We, we didn't really have any control over what got treated and what didn't get treated.
Thank you.